Good afternoon, everyone, and welcome to the second in the ABRF sequencing seminar series. We're delighted today to host and welcome our colleagues from Illumina. But before we get started, I just wanted to share a couple of announcements and updates from ABRF. In particular, we just want to remind everyone, please feel free to introduce yourself. We're going to use the chat for all of this, so your name, where you're calling in from or connecting in from today, your institution. If you have any questions for our presenters, we're going to also use chat for those. So feel free to handle those, to post them there. Presenters will address those questions following their remarks. Today's session is being recorded, and the recording will be available and shared with all the registrants following the program, and it'll also be posted to the education section of the ABRF website. Just wanted to share with you a couple of upcoming events. We have the remaining sessions in this series, continuing pretty much weekly with we're skipping a week for one of the upcoming meetings. And you can see the list of companies that'll be presenting. Same time, same day of the week, each week, 2 p.m. Eastern. And you can see the link to register for any of those programs. ABRF chapters are also hosting several events in the next few weeks, and we wanted to share those dates with you. Next week, in fact, September 21st and 22nd, our West Coast chapter will be hosting their meeting at UCLA, and you can see the keynote speaker featured there. And then in October, we have 2 chapter meetings. Rochester, the Northeastern chapter, will host their meeting October 12th and 14th. Registration is open and still available. And finally, the Midwest chapter, the following week, October 17th and 18th, they'll be in Columbus for their meeting, and you can see the theme for that meeting. And then lastly, it's not too early to be thinking about the 2023 ABRF annual meeting. Registration for the meeting will open in October. And just for this audience, I can share a little bit of an internal secret. We are planning and very close to finalizing arrangements to host the closing event for the meeting on May 10th at Fenway Park in Boston, the historic landmark and home of the Red Sox. So whether you're a Red Sox fan or not, it's a wonderful venue, and we're really excited about hosting the annual meeting there. So with that, I will stop sharing and turn it over to Sam and Fiona. Thank you.
Thanks, Ken. Hi, everyone. Let me get my screen shared really fast. Just wanted to confirm if you guys can see this. Awesome. All right, so we'll get started. So hi, thanks for joining us today for some updates on the capabilities of Illumina sequencing platforms. I am Sam Hester, product marketer for Benchtop systems here at Illumina, and I'm joined by Fiona Nohili, who's the manager for product marketing. And we're thankful to ABRF and your community for giving us the opportunity to come today and share some of these updates and recent additions. So we're really excited to be here. Let's just jump right in. So as many of you know, Illumina has made strides in the past, not only to provide excellent and accessible sequencing technology, but to bring that accessibility and performance to every part of the sequencing workflow. When considering our product portfolio, we wanted to keep in mind our customers and how products are used in order to make flexible and comprehensive solutions as simple as possible to achieve. And today we wanted to just share some recent product updates in each of these 3 major categories, from library prep and sequencing through data analysis and interpretation. So starting with library prep, I wanted to introduce a couple of newer improvements we've made to 2 popular workflows, exome sequencing and microbial identification. So let's start with exome sequencing. Exome is a popular solution due to the time and cost efficiency compared to whole genome sequencing, while still providing a broad look at the coding region of the genome. And it provides manageably sized data sets for analysis while still being comprehensive enough to be able to replace many smaller panels or single tests. So Illumina has partnered with Twist Biosciences to provide a more extensive exome solution with our new Illumina DNA Prep with Exome 2.0 Enrichment Kit. This new exome panel uses our Illumina DNA Prep segmentation chemistry, which overall significantly improves the speed as well as the hands-on time needed for this workflow compared to some of our older kits, including the TrueSeq exome kit. And at launch, we've had multiple sample input types available, although it's important to note that we're still working on FFPE. The panel itself has been updated to provide extensive coverage of targets across current public data sets, which we'll show in a second, and is manufactured under ISO 13485 compliant process with Twist. And most importantly to us, we wanted this new panel to represent a complete solution for finding answers through exome sequencing, not just to be a piece of the puzzle. When using this library prep and enrichment reagents, after sequencing, we have the DRAGEN enrichment app for secondary analysis and variant calls. And then we have Illumina Connected Insights Germline for downstream tertiary analysis, bringing you all the way from extracted DNA to functional data. There's also going to be automation scripts available at launch based on our current Illumina DNA Prep with enrichment scripts, although there are a few modifications needed for Exome 2.0. So here's a closer look at the workflow that I mentioned previously. It's nearly identical to our existing Illumina DNA Prep with enrichment workflows, split into two parts, library prep and enrichment. The overall workflow taking about 6.5 hours, and there will be 2 total hours of hands-on time split between the library prep and enrichment portions. Again, the major time saver over our older TrueSeq enrichment stuff was the tagmentation piece, which significantly reduces that first step and makes this the fastest exome sequencing solution that we have. So getting into some of the data, I wanted to start with this slide showing the coverage of the new Exome 2.0 panel compared to 2 of our previous Exome products that we've had. You can see that the Illumina Exome panel, sometimes called IEP, and then sort of a recent Illumina Exome V2 panel produced through IDT. We're comparing both of those to our new Exome 2.0 panel with Twist. And so Twist will be the dark orange bar. So this is an in silico comparison that is using bioanalysis only to show coverage of all 3 panels across 10 public data sets. And what you can see is that in 7 of the 10 data sets, the new 2.0 panel shows the best coverage, in some cases by a wide margin. There were only a couple data sets where 1 or both of the old panels showed slightly better coverage, but overall the new panel shows a marked improvement. It's important to note that right now the Exome 2.0 panel doesn't include mitochondrial content off the shelf, but there is a spike in mitochondrial panel available from Twist that partners very well. And our team is really happy to work with you to get this implemented if this is something that you're interested in or require. Moving from in silico to real data, this data set was generated using downsampled libraries at 50 million reads with 24 replicates in a per hybridization condition. It was either a 1.5 hour hide compared to a 16-hour overnight hide using the NA 12878 genomic DNA sample, because that's very well characterized. And here we were looking at what percentage of the targets were covered at greater than 20x for each of these data sets. And you can see clearly that there's near complete coverage of all of the targets in these data sets for both hybridization conditions, but more specifically, the 1.5 hour hide was nearly uniform and consistent with coverage with the 16-hour overnight step. So shortening the time for your workflow really doesn't affect your results. Although if you do, end up not finishing the workflow in one day and need to leave it overnight, that will actually make both consistent, but also slightly better improvements in your data. Next, we wanted to look at how our newer 2.0 panel performs across some standard metrics compared to our now older original exome kit designated IEP here on the right in the gray bars. You'll see that a padded enrichment was really good with over 90%. Pad and base enrichment was nearly 90%, and mean target coverage was also really good with 62x for 1.5 hour hide and nearly 70x for the 16-hour hide. And again, this was used, this was at only 50 million reads per sample, not the standard 100 million reads. Moving from in silico to real, that didn't change my wording. I went back. I think that's what happened. There we go. Sorry about that. Anyway, continuing. On the left, you'll see uniformity of coverage for the new 2.0 panel has some improvements compared to our current, actually now older panel. And in the middle, you can see that there's a much higher fraction of the targets that are at least covered at 20x. On the right, you can see that when looking at the fraction of targets covered at 50x, again, with only 50 million reads, that the percentage of the 1.5 hour hybridization is comparable to our older kit, and the overnight hybridization actually increased that to 62%. Of course, when you look at a higher coverage of targets, the fraction of targets covered at that level is going to decrease, which is what's shown on the next slide. That is the graph on the left, the fraction covered at various depths. And as you can see, again, there's an improvement of the Exome 2.0 panel in coverage as depth increases compared to the original Illumina Exome panel. In the middle, the fold 80 base penalty is intended to show how to improve the graph on the left in terms of how many additional reads do you need across the set of bases to improve target coverage. And a lower score is generally better here. And you can see that the new panel is showing significantly older and or significantly improvements over the older panel here. And then finally on the right at the fraction of targets at zero coverage, being an Exome panel, this is a very large panel. There are over 300,000 targets. So there's always going to be some targets that are missed, but you can see that the new panel misses quite a bit less than the existing Exome panel. And even, given the sheer volume of targets, even a small percent improvement here is really substantial. All right, wrapping up the Exome, we've discussed the library prep and data extensively, but to touch on other parts of this workflow, this works well with any of our mid to high throughput sequencers, and you could fit up to about 430 or so exomes on a NovaSeq S4 run at 50x coverage for reference. DRAGEN Enrichment app provides rapid secondary analysis for variant calling, including CNVs, and is available both on-premise as well as a base-based sequencing hub. And finally, you can achieve tertiary interpretation with the Illumina Connected Insights germline analysis for high-powered variant annotation filtering and visualization. All right, so next I want to take some time to discuss our respiratory pathogen ID AMR panel called RPIP for short. This is a microbial identification panel designed to target lower respiratory tract infections of multiple types of pathogens, as well as detection of antimicrobial resistant genes. It's meant for rapid detection and enables a very fast turnaround time from sample to answer, in some cases less than 24 hours. What really makes the RPIP panel so powerful, however, is its breadth. It can be used as a single test to efficiently provide answers that labs traditionally have had to run multiple tests over many days to achieve. Even more of a couple of these tests shown might be needed to detect a co-infection or something like that. For example, RPIP can save significant time and cost by providing that range of detection into one single test that's very fast. Diving more into that depth and breadth, this slide effectively summarizes why RPIP, really allows that precision metagenomics of a wide array of potential pathogens. There are over 180 bacteria, over 40 viruses, over 50 fungal species that can be identified, including full genome coverage of SARS-CoV-2, as well as the flu viruses, or to a few different flu viruses, I believe. In addition, there's extensive AMR detection with more than 2,000 resistant markers included. So this is really truly is like a very broad panel of detection. In terms of recommended sequencing, you can see the array of our low and mid throughput portfolio has options to run various degrees of samples for this kit, but there's two in particular that I would recommend. At the top, you can see our MiniSeq rapid flow cell configuration can handle 20 samples of RPIP with 1 million reads each in just 5 hours of sequencing time, which combined with fast library prep and really fast analysis allows you to get that sub 24-hour answer from sample to result for pathogen detection and antimicrobial resistant gene detection. At higher throughputs, the NextSeq 1000 and 2000 at the bottom is going to enable faster runtimes and much better pricing than the NextSeq 550 for the same number of samples. Not shown here on this slide, but we're covering it elsewhere in the presentation. We have our new P1 100 cycle kit. That's coming soon in Q4, and it's going to allow the NextSeq 1000 and 2000 to run about 96 samples here for a really cost-effective and fast intermediate option on this cart. On this chart. And we'll get to that kit in just a bit. If you're going to have some more questions about it, we have a few more answers later in the presentation. And then for analysis, we have the Explify RPIP app on BaseSpace. Oop, did I mute? No, I didn't. Sorry, I hit a button. This provides a PDF report of our text-based report that includes data QC, detected pathogens, consensus sequence for SARS-CoV-2 and flu viruses, as well as AMR resistance. You can see an example report on the right that detects polymicrobial pneumonia with some AMR resistant genes detected. And this represents a detailed ability to detect this outcome much, much faster than a traditional method would have been to do the exact same thing. And finally, just some data that shows the performance of RPIP with ID by DNA Explify analysis, identifying all pathogens, even those at a low copy number. All right, we're going to transition a bit to our sequencing portfolio or the updates there. Just calling out in general, we've been developing our sequencing platforms for a long time. It's been well over a decade now since the HiSeq or the introductory MySeq has come out, and our latest sequencer, the NextSeq 1000, 2000, came out just a couple years ago, and we are still continuing to develop improvements on that platform. Our goal at Illumina has really always been to have solutions for every customer, no matter what type of project or what level of throughput that you're at. We're hoping to have a solution across our portfolio for all of those things. And as you can see, some of our later instruments, such as the NextSeq 1000, 2000, the NovaSeq, et cetera, we've really striven to have a flexible system that can achieve a really wide variety of methods and applications that are possible on that system. Since the release of that NextSeq 1000 and 2000, we've been continuing to update and improve it as we can. We're definitely committed to continuing to do so on this newest platform. You can see that, really at release, we really only have the P2 flow cell. And then after a short time, we came out with further flow cell options in terms of P3 at the end of 2020 and P1 at the end of last year, the end of 2021. We've had additions to the DRAGEN onboard analysis platform. We've had addition of custom primers and recipes available, improvement in our output and Q30 specs, and extensions to our shelf life. So we've been really working hard to improve this product. And so we've been really happy to announce that we're going to be continuing to do so, including introducing longer reads capable on the NextSeq 1000 and 2000 platforms. If you guys were able to check into AGBT earlier this year in June, we did talk about this new P1 flow cell configurations of both 100 cycles and 600 cycles coming to P1. And now here today, we were really happy to announce that we are having, in addition to these two kits coming in Q4, a third kit is coming in Q4 as well, a P2 flow cell with 300 million clusters capable in flow cell, also at 600 cycles or a 2x300 base pair read. And so this is super exciting. We think this is going to add a lot of capabilities to this platform, and we're really excited. So diving into those a little bit, here's the total NextSeq portfolio that's going to be available once these kits are released. You can see the full flexibility of the platform. We're adding new flow cells to both P1 and P2, creating a really wide range of outputs and throughputs that are available, and really cementing the different types of read length configurations that you can do. Adding in P1, it was just 300 cycles. Now it would be 100, 300, and 600 cycles that you could run with 100 million reads. And then at P2 with 400 million reads, you can run 100, 200, 300, or 600. Although to caveat, the P2 600 cycle kit will be at 300 million reads, not 400. What we believe this is going to do for the NextSeq 1000 and 2000 platform is really extend the capabilities of what types of applications, methods, and types and use cases that the NextSeq 1000, 2000 will unlock for the user. In addition to what we've been able to do, including, like targeted panels, exome and large panels on the larger kits, RNA-seq single cell, even liquid biopsy. On these longer read kits, we really believe that this is going to add capability of small whole genome de novo assembly, targeted metagenomics, HLA typing, shotgun metagenomic capabilities, metatranscriptomic capabilities, and immune repertoire profiling. And so this is going to be a great addition. In addition, the P1 100 cycle kit is going to help out with smaller pilot projects, batching needs on RNA-seq or single cell projects on a lower throughput scale. Calling out, since we have all these kits really fast, we have color coded these kits since we have so many now capabilities for the NextSeq 1000 and 2000. We believe it's really important to help the user with storage and organization of these flow cells to help you recognize them in the lab really quickly. So we have updated our colors and labels on these, compared to our past sequencers to make this more user-friendly in your lab. And then most excitingly, I just want to look at some examples of, just like how this will affect our portfolio and our use case, and some more specific examples on these newer, longer read kits. And so, we could see this chart here. A lot of these capabilities were already on the NextSeq 1000 and 2000, including single cell and mRNA, which used to be, really popular applications and still are on the P2 and P3 flow cells. But now on the P1 flow cell as well, you're going to be able to run smaller amounts of samples, like one sample of single cell or a few samples of an RNA-seq project on a P1 flow cell to do, pilots, interesting samples, things like that, before you scale those up to your larger studies. Immune repertoire sequencing is something that we're really excited is coming to the NextSeq 1000 and 2000 as well. At sort of heavier depths for immune repertoire sequencing, they have been really limited on past technologies due to the level of throughput. And so at higher levels of depth, such as 5 million reads per sample, you'll be able to achieve 20 or 60 samples on these new P1 and P2 flow cells, which is going to really unlock a lot of projects and ability to discover things using that application. One of the most important things to our customers, although we don't have all the data that we can release just yet, is concordance with these new longer read kits with our previous MiSeq 600 cycle kits that have been out for a long time, and they're one of the most popular kits to use on one of our oldest and most popular platforms. And what we've done is we've looked at quite a few internal studies to kind of make sure that the kits that we are providing with the MiSeq provide additional speed, higher output, better pricing, but at the same level of expected performance or better. And so that's what we're trying to show here, is that the NextSeq 2000 600 cycle kits are able to achieve the same level of bacterial identification in a 16S project than those same 600 cycle kits on the MiSeq, and that there's strong concordance between those two kits in terms of microbial representation, so that you guys could be comfortable switching over your projects and unlocking new projects with higher throughput and better pricing on these new kits. At the same time, we really believe this is going to sort of unlock things, as I mentioned, within immune repertoire profiling, because there's a much higher throughput at a better cost compared to MiSeq. You can run projects at greater depths with and less input amounts. And what we've shown here is that in terms of compared to MiSeq, you can run much more samples and identify more total clonotypes on the new NextSeq 1000, 2000 P1 and P2 600 cycle kits than you could previously on the MiSeq. And then, of course, I did want to call out an update to Onboard DRAGEN on the NextSeq 1000 and 2000. In case you're not aware yet, the license for Onboard DRAGEN on the NextSeq 1000 and 2000 is now free of charge. It comes with your purchase of the system. If you already have a system, it should now currently be free for you. And it comes with multiple pipelines on board, including single cell, enrichment, RNA, germline, amplicon, or a compression, FastQ. And we are still looking to add more pipelines to DRAGEN Onboard NextSeq 1000, 2000. If you have a favorite pipeline that's not included, definitely reach out to us and let us know. It's currently available on the rest of the pipelines are on base space, but we're trying to expand this as much as possible to make this an easy use case for you. Especially for that Exome prep that I mentioned earlier, DRAGEN enrichment is onboard NextSeq. So you could, for example, use that prep with our newest Exome panel, use the DRAGEN enrichment onboard DRAGEN here, and then go straight to tertiary analysis off of your sequencer. So I think this adds a lot of capabilities to the NextSeq 1000 and 2000 systems, and there is definitely more to come. That's all I have for my section. I'm going to go ahead and stop sharing now and pass things over to Fiona.
All right, thank you, Sam. I'm going to go ahead and share my screen here. And hopefully we can get in presenter mode. Sam, are you able to see my screen there in presenter mode?
I can.
Okay, let me minimize myself here. All right, well, hi everyone. I'm Fiona Nohley and I'm part of our AMR product marketing team. And I wanted to touch on a few updates on the informatics side, as we've been busy on updates across the portfolio, as you can see, but I wanted to touch on a few updates that we've had in the last couple of months on the informatics portfolio. So here's sort of a broad look at what we have for the Illumina connected informatics portfolio. And we have things that happen upstream of the sequencer, like Clarity LIMS and Local Run Manager, Base-Based Sequence Hub. But today we're really going to kind of focus on what happens after the sequencing is done and what you want to do for secondary and into tertiary analysis. So we'll talk a little bit about updates to our DRAGEN secondary analysis pipeline, and then we'll touch on Illumina connected analytics as well. So DRAGEN is our secondary analysis pipeline, and we've made a lot of improvements. And as Sam mentioned, DRAGEN Onboard NextSeq 1000 and 2000, but the premise of this solution is to meet you where your data is, so that if you need to have a local analysis solution, you can utilize an on-premise server or onboard the NextSeq 1000 and 2000. If you have the capabilities to go to the cloud, we have DRAGEN that you can plug into within Illumina Connected Analytics or Base-Based Sequence Hub for that secure cloud analysis. And when DRAGEN, we first acquired Edico a few years back, I think that we really were focusing on the speed and efficiency of DRAGEN. But DRAGEN is much more than just being able to analyze a genome or an exome really, really fast. It actually does it very accurately. And we've been continuing to build out a comprehensive suite of solutions that are powered by these built-in FPGA cards on board the DRAGEN server itself or within the cloud instances that you can utilize DRAGEN either in BaseSpace, ICA, or with your own AWS instance. So we'll talk a little bit about the updates that we've made on accuracy. We'll talk about the updates to comprehensiveness, the different offerings we have for different DRAGEN pipelines, and then we'll talk about the speed because that has been improved. And there's also some pieces of that are related to lowering costs with data compression. All right. So speaking about the accuracy, and you may have seen this before, so it's not that we're just continuing to show the same slide. However, we did want to make some important updates here as we did release DRAGEN 4.0 earlier this summer in July. So this slide was shown sort of a version of this earlier this year, probably at ABRF meeting itself. But what we've done now is enabled the DRAGEN graph reference and Illumina machine learning by default on the 4.0 software. So this allows, if you're looking at this data, it's looking at all benchmark regions that were done through the Precision FDA Truth Challenge. And this is looking at benchmark data across all those benchmark regions and showing a 99.83% score for that accuracy that you can see that's shown by the F1 scores. So if you're combining Illumina sequencing, which is shown in orange, with the latest version of DRAGEN 4.0, enabled by default are these accuracy improvements to get you to essentially equal or better offerings for accuracy as compared to other sequencing platforms and analysis options. And then if we look at some other difficult regions, so right now this graph is looking at the MHC region, which is highly repetitive and often difficult to map. We see some of the same trends holding true with Illumina sequencing data combined with the DRAGEN 4.0 graph. You can see that has the same or better accuracy as shown by the F1 scores across different sequencing platforms and data analysis types. So this is the combination of the machine learning that is enabled, as well as the DRAGEN graph that gives you this ability to have the most accurate reads and even in the difficult regions. And so kind of taking another look at the comprehensiveness, this is showing sort of what underlies all of the different DRAGEN applications. So within the context of mapping aligning, we have several different pipelines that can be enabled on premise on your DRAGEN server or in the cloud through ICA or BaseSpace. You can see sort of in the germline category, there's lots of different callers to call SNVs. There's Expansion Hunter for things like SMA, there's CYP2D6 callers, HLA, GBA, that's sort of on the germline side. And then if you take a look at the somatic side, we have tumor normal callers, tumor only, and then different types of biomarker callers like TMB and MSI that are important in oncology research and testing areas. And then we've also expanded on the RNA front. We have different bulk RNA analysis pipelines for fusion detection, expression, and we now have a single cell RNA pipeline. And we also partnered with NanoString to be able to package the data that comes off of a NanoString. If you're using the NanoString geomics assay and running it on a Illumina sequencer, you can then move that data into BaseSpace and package it in a way that is a little bit easier for you to then go into tertiary analysis using this DRAGEN geomics app as well. And then when we talk about efficiency, we've talked about speed in the past, and I have some data to show some of the improvements that we've made with the latest 4.0 version. But when we talk about efficiency, we're also thinking about data compression through our Aura technology, which was enabled by the acquisition of Anancio. And then just the efficiency with the combination of the speed and the compression, you're now reducing the file sizes and enabling a faster transfer time for those larger files that typically can take a while to move from one place to the other. And so here's sort of the latest in terms of the analysis time. You can see here on the chart here, compared to sort of open source methods, we have for small variant calling, it's down to 17 minutes for mapping, aligning, and small variant calling for sort of a 34x or in that range human genome. And then if you're looking for sort of the full suite of all different variants, that would be in the range of 30 minutes. And you can see how that compares to the open source tools. And then in terms of being able to kind of reduce those file sizes, we know that this is something that is constantly a challenge, and we're working to improve this through our DRAGEN technology and the acquisition of Edico and their compression technology. And you can see even comparing when you compress that file, you get a fastq.aura file, and that is about 5 times less than your fastq.gz, which just enables you to transfer that a little bit faster and hopefully also reduces the amount that you have to spend on storage. And so this is sort of a kind of a recap of all the different things that we've done. I think one of the things that I wanted to kind of call out is just the sheer number of updates that happen across the DRAGEN platform. And it can be a lot to keep track of. So I fully appreciate that, even as it's part of my role to kind of keep track of these things. There's often multiple updates within the year, which is just a testament to how much we're improving across these different areas of efficiency, comprehensiveness, and the accuracy scores. But sort of what we've enabled here with the recent versions 4.0 as well as the 3.10 that we updated, I think was maybe toward the beginning of this year, was DRAGEN machine learning enabled by default. We had some PGX targeted callers, imputation, and then we've added the additional single cell ATAC-seq app as well. And you can see that these updates sort of correlate nicely with the amount of genomes that are processed, or let's call it whole genome equivalent. So we actually passed the over 1 million whole genome equivalents in 2021. I think we're excited to see sort of where we get to this year as well. But we see sort of a tremendous increase in the amount of data that's being generated in general, but also the amount that's being processed by DRAGEN. And we're just kind of at the beginning of this, and we're still looking to make improvements across the next several quarters and years. Okay, so shifting gears a little bit, sort of still within generally the category of secondary analysis, but ICA also gets into tertiary analysis. We have Illumina Connected Analytics, which we officially launched at the beginning of this year. And this is a cloud-based platform that allows researchers to do lots of different things. So you can do analysis at the experimental level. There's opportunities to do population genetics. And then for those that are trying to do clinical research, there's some opportunities to kind of aggregate data and share data across different work groups. So a few of the sort of key features of ICA are the ability to build and customize analysis pipelines. So where BaseSpace was our previous and sort of legacy cloud-based analysis solution, it was really locked down into sort of these GUI, graphical user interface, easy to use apps, but it did mean that you couldn't really customize things within it. And I think that we understood that and wanted to build a platform that would allow you to do that customization that is needed across different types of secondary and tertiary analysis. And then we also know people need to do this at scale. So if you're scaling out a lab, you want to be able to kind of lock down some workflows, productionize those, and then allow those to be perhaps in the end user's hands to be able to do some of the more basic tasks and allow your bioinformaticians to kind of be freed up to do more of the complex tasks that may not be as straightforward. And then being able to explore and share that data is sort of a key tenet of ICA. And so within the building of custom pipelines, there's different tools that are available, Nextflow, Docker, there's CLI, command line interface, as well as RESTful APIs that allow the customization of these different pipelines. There are DRAGEN pipelines that are sort of pre-built into the ICA platform, and these can then be customized to the needs of the particular workflow. And you can go through and kind of lock down if there's any sort of need to have automation for different validated environments that could be enabled on this as well. And so just sort of another look at kind of different ways that ICA can be utilized. If you take a look at sort of the top part of this chart, we sort of see, the sequencer and data coming off the sequencer, and ICA fits within secondary analysis into some components of tertiary analysis, but there's still maybe other downstream analysis pipelines or software you may want to use following the analysis in ICA. And so you can use this at a single sample or population level. And we'll go into that in just a second here. So for the single sample, and this could be sort of probably a little bit more, maybe not in the academic core lab realm, but sort of you can imagine different opportunities for this outside of, this example, sort of thinking about rare disease, where you might be having one sample or a trio that you want to run through this certain pipeline that's locked in for rare disease diagnosis. This would be utilizing some of the DRAGEN pipelines for secondary analysis, sample QC and review, and then that data could be packaged into what could go into TrueSight software suite into a report for those kind of single sample analysis workflows. But then there's lots of other kind of utilization of multiple other workflows in parallel. If you're doing lots of different samples, you can run your DRAGEN pipelines or different other custom pipelines for secondary analysis and move into our Jupyter notebooks and interactive analysis offerings to then kind of customize where that data comes from DRAGEN or another app that pulls into and sort of allows you to aggregate and look at multi-omic data within ICA. And so that concludes this section here. We talked a little bit about DRAGEN and ICA. There's a lot more sort of that we're working on across the connected informatics portfolio, but just wanted to kind of touch on a couple updates here. So with that, I actually have one more slide, I believe. We just wanted to highlight the Illumina Genomics Forum. This is happening actually in about 2 weeks in San Diego. So you've probably received a few emails on this. We've been sending out a couple, and there will be sort of a number of different agenda topics and speakers. There's one session that's the Illumina Innovation Roadmap that I think would be of particular interest to this crew. So if you're able to make it in person, there's still opportunity to register now in person in about 2 weeks, and then there will be sort of a virtual recap that happens on October 4th. So keep a lookout for more information on that. I would strongly suggest listening in to this, and we're happy to take any questions at this point. Thank you for your attention.
All right, there were a few in the chat, so I'll just read those out to start. The first one will be updating colors and packaging on other platforms and kits. I do think that we are kind of committed moving forward to making sure that all new kits and new platforms have this sort of color coding. We've heard really positive feedback about it. In terms of whether this is coming to older kits, I don't think there are plans for that right now, but I don't think it's off the table. And if you feel strongly that that's something that you definitely need, do reach out and provide your feedback to us, and we'd be happy to pass that along. There's another one. Is Illumina going to sell new library construction kits for the 600 base pair flow cells? I do believe that our 600 base pair kits will be compatible with all of our current library prep that we have right now and that might come out. There's no reason I think that there would be any new library prep kits needed to be run with them. They should be fully compatible at launch. And then one for you, Fiona, can we work with novel genomes and transcriptomes on DRAGEN?
Yes, so DRAGEN was initially built sort of with human whole genome sequencing in mind. That said, we've obviously made a number of improvements. I think it might be good to kind of check on if there's specific species that are of interest. We may want to kind of chat about that directly to see. I may have to follow up on that one, but it's something that we're looking at improvements. I will say it's sort of the focus is on whole genome. It was initially on whole genome sequencing and sort of other human related applications. But it's certainly something we've looked into, and I'd love to kind of follow up offline on that.
Okay, next also for DRAGEN, can you speak a little bit more on the GeoMx integration with DRAGEN?
Yeah, so that one's relatively new that we've come out with, and there's sort of two options for you to be able to access the DRAGEN GeoMx app. There's one on board the NextSeq 1000 and 2000, and then the other is through BaseSpace. And it essentially it helps on two fronts. So at the beginning, when you're setting up your run, it sort of is allowing you to say that you're running a DRAGEN GeoMx, or sorry, a GeoMx assay on the NextSeq, and then it will help on that run setup piece. And then it also, if you want to do sort of the part of the analysis before you go into NanoString suite of solutions, it sort of packages the data that makes it a bit easier to plug into the NanoString data analysis solutions. So it's not necessarily replacing that, but it is sort of augmenting and allowing it to be a little bit more seamless for run planning and that initial kind of packaging of data to get it over to the analysis solutions for GeoMx.
Okay, I'm going to bundle the next two together because I think they'll have a similar answer. Do you have any more information regarding Chemistry X and existing instruments it will be implemented on, and if any? And then any information on Infinity long reads and maybe when we can expect to hear more about it?
Yeah, I think I'll go back to this slide and just say that you should, as much as you can, tune into this event or attend. We are going to be talking about our innovation roadmap and sort of following on what we had announced at JP Morgan in terms of Chemistry X and Infinity long read technology. So there is more information coming soon. And that's all I could say right now, but I would keep your eyes peeled around September, the week of September 28th, and be in touch with your local account managers as there's more information coming soon.
Do we have any plans on staying price competitive or on par with vendors such as Singular and Element?
Do you want to take it, Sam, or do you want me to?
Yeah, I think that, look, we're definitely considering price as an important aspect of our sequencing workflow. I don't know that we have any definitive plans, in place or ready to be in motion right now, but I think Chemistry X in particular is one where we're looking at where we can make improvements to our sequencing technology to lower costs. Will we be able to use DRAGEN or ICA with data generated from other sequencers?
Yes, I mean, the short answer is yes. I think from an Illumina standpoint, we wouldn't necessarily be working on enabling that, like working directly with you to enable that, but there should not be technical limitations to that, as if they are BCL files similar to the output that comes off of our sequencers. There is not limitations on the technology side of things to be able to use that for FastQ or other apps within DRAGEN for similar types of data files.
The next one is a question / suggestion, and I can take it. Can Illumina offer shorter read lengths on P1? I definitely think that's a great idea. I know that we're looking at more kits on the NextSeq 2000. We're not done with it, even after these 3 kits. So all suggestions are on the table. We're looking at what we can do, and I think that's a good idea. That's all in the chat for now. Are there any others?
I will say, just as we're waiting, if there are any more questions, I'll just put this up here. If you have any follow-on questions or feedback you want to share, this is Sam and my emails. Please feel free to reach out to us directly to provide feedback. We're here to listen to all of it and take it in, as we really appreciate the group at ABRF and making sure we are listening and are here. And thank you for having us at this webinar, but please feel free to ask more questions. We have some more time, but if you have other things you want to chat about offline, you can reach out to us directly as well.
Nothing new popping in. I think we are
okay. Well, thank you both very much. We really appreciate that informative update. For those on the line, we have another seminar coming up next week. As I said, same time, same day of the week. Please join us for another update. So we appreciate everyone, and thank you both very much to our speakers from Illumina. And thank you, Anoja. Have a great day, everyone.
Thanks, everyone.